Start one.
Start.
Hello everyone. Good morning. My name is Dhairya Trivedi.
My name is Vij Desai. So today we'll present on Parkinson's disease and the physiological aspect of the Parkinson's disease. So let's start. Invisible. Dhairya, can you check?
Yes, yes, yes. Screen is visible.
Okay. So today we'll learn about the mysteries of Parkinson's disease. So Parkinson's disease is a neurodegenerative disorder characterized by progressive loss of motor control. So understanding the intricate mechanisms underlying the disease is crucial for developing effective treatments and improving the quality of life for millions affected worldwide. This presentation explores the multifaceted nature of PD or the Parkinson's disease, delving into the physiological processes that contribute to its development and progression. So as you can see in the image, sorry, yeah, this is the substantia nigra of the healthy patient and the person with PD or the Parkinson's disease. And you can see how the substantia nigra is clear in PD and in the healthy one. And this is the putamen and this is the caudate nucleus. So we'll get it further. Okay. So the first aspect is the neuronal loss or the silent killer. Dopaminergic neuron decline. The brain's intricate communication network relies on specialized cells called neurons, as you all know. And dopaminergic neurons, which produce the neurotransmitter dopamine, are crucial for movement control, mood regulation, and cognitive function. In PD, these vital neurons undergo progressive degeneration, disrupting the delicate balance of brain signaling. So, the dopaminergic neurons are essential for the movement and mood regulation, and this declines in the PD patient. So, substantia nigra, the epicenter, the part which is in the midbrain region, densely populated with dopaminergic neurons. These neurons extend their projections to the stratum. Stratum is the caudate nucleus and the putamen, as you all can see. So, these neurons extend their projections to the stratum, a key brain area responsible for initiating and coordinating movement. The loss of dopaminergic neurons in the substantia nigra leads to the hallmark motor symptoms of PD, which are tremors, rigidity, and slowness of movement. So, the mechanisms of neuronal death. The neuronal death in PD can occur through various mechanisms, including apoptosis, which is the programmed cell death, as you all know, and oxidative stress. Apoptosis is a regulated process where cells self-destruct while oxidative stress involves damage to the cellular components caused by an imbalance between free radicals and antioxidants. These mechanisms can be triggered by a cascade of events, ultimately leading to a neural loss. So this you can see substantia nigra and the healthy patient and the PD patient. The neuronal or the neurotransmitters which are going from one neuron to another. So this is the postsynaptic terminal, and the dopamine is very less going in the PD patient, while in the healthy patient it's going effectively and more in quantity, as you all can see clearly. So this is the neuronal loss which happens in the PD patient. So the next point is the protein misfolding, a delicate balance. So alpha-synuclein, a building block of the brain. Alpha-synuclein is a protein found in the brain that plays a crucial role in the normal function of neurons. It helps in the formation of synaptic vesicles, which stores and release neurotransmitters. However, when alpha-synuclein misfolds, it can become toxic to neurons. Misfolding, a chain reaction. Alpha-synuclein misfolding occurs when the protein structure becomes altered, leading to aggregation and clumping. The misfolded alpha-synuclein can then spread from cell to cell, leading to the formation of Lewy bodies. Now, what are Lewy bodies actually? The mark of Parkinson's. Lewy bodies are aggregates of misfolded alpha-synuclein found in the brains of individuals with PD. These protein clumps disrupt normal neuronal function, leading to communication problems and ultimately contributing to neuronal death. So, what's the impact on neuronal health? The formation of Lewy bodies and the spread of misfolded alpha-synuclein can disrupt the delicate balance of neuronal communication, This can lead to impaired signaling, reduced dopamine production, and ultimately neuronal dysfunction and death. So, as you can see, sorry, yeah, um, as you can see here, these are the vesicles, as you can see here, synaptic vesicles, which transfer the dopamine from one neuron to another. And as I mentioned, in PD patients, there are very less because of the protein misfolding, and alpha-synuclein is responsible for that. So misfolding occurs, and therefore there is a less spread from cell to cell, and less dopamine is released. So it's a key, and Lewy bodies are formed eventually. So this is the mark of Parkinson's. Yeah. So as you can see, this is the clumping, and the neurotoxicity occurs in the neuron. So this is the mitochondrial dysfunction that is also responsible for the Parkinson's disease, because mitochondria plays an essential role in powering the brain. So mitochondria, it is the powerhouse of the cell, as all of you know. Mitochondria are essential organelles within neurons responsible for generating energy through cellular respiration. They convert glucose and oxygen into adenosine triphosphate, the primary energy currency of cells. Mitochondrial damage, disrupting energy production. In PD, mitochondrial dysfunction is implicated in the loss of dopaminergic neurons. Mitochondrial damage can be caused by a variety of factors, including oxidative stress, genetic mutations, and environmental toxins, which we'll learn further. Energy deficits, the struggle for survival. Impaired mitochondrial function leads to decreased ATP production, creating an energy deficit in neurons. This energy shortage can disrupt vital cellular processes, ultimately contributing to neuronal death. So it is very, very essential that mitochondria should be present in a large quantity to make the cellular processes. The interplay with oxidative stress. Oxidative stress and mitochondrial dysfunction are intricately linked. Mitochondrial damage can lead to increased production of ROS, reactive oxygen species, which are damaging free radicals. This ROS in turn can further damage mitochondria, creating a vicious cycle. So this cycle is vicious, and therefore it weakens our brain and causing PD. So as you can see the picture of the healthy mitochondrion, the damaged mitochondrion. Let's move further. Neuroinflammation, the body's defense. So as you're all aware, microglia, the brain's immune cells, are specialized immune cells in the brain that play a crucial role in protecting against infection and injury. They act as scavengers, engulfing debris and pathogens to maintain brain health. Chronic inflammation, a silent threat. In PD, chronic neuroinflammation is believed to contribute to neuronal damage. The presence of misfolded alpha-synuclein and other toxic substances triggers microglia activation, leading to a prolonged inflammatory response. Neuroinflammation and neuronal loss. Chronic inflammation can have a detrimental effect on neurons. Activated microglia release inflammatory mediators that can damage neurons directly and contribute to the progression of neuronal loss. Therapeutic targets, renin and inflammation. Targeting neuroinflammation is an emerging area of research in PD. Drugs that reduce inflammation may offer potential therapeutic effects by protecting neurons from further damage and slowing disease progression. As you all know, there is no treatment for PD, but we all can do, like scientists are working on the targeting neuroinflammation techniques. So they target the neuroinflammation, and it can reduce of inflammation, which offer potential help in the PD. So as you can see, as you can see, the resting microglia and the activated microglia. And if the resting, this is the process of microglial cell, and the cell becomes, activated more and more, and it is bad for our neuron. So neurotoxicity happens, and which causes PD. And sorry, our image was actually misplaced in the PPT. So these are the misfolded proteins. I again explained it. These are the misfolded proteins. These are the oligomers, and these are the fibrils, which therefore contributes in the formation of Lewy bodies. So yeah, this was the protein misfolding, alpha-synuclein, which I was talking about earlier. So yeah, now I think Darya Trivedi will proceed with the presentation. Thank you, ma'am. Start there, Trivedi.
So thank you, Dvij, for the explanation. I
You're
will do
welcome.
further explanation. The chemical imbalance and the disrupted communication. The neurotransmitter, the dopamine. The role of dopamine is to control movement, mood, and motivation. Their impact in PD, decreased level lead to the motor symptoms. Glutamate. Their role is the excretory neurotransmitter, and the impact in PD is the excessive glutamate activity can be toxic to our neurons. Third, GABA. It is the inhibitory neurotransmitter. Their impact in PD is that they can reduce the GABA levels, which can contribute to the neural excitability. The genetic factors. The SNCA gene. The SNCA gene encodes for alpha synuclein. Mutation in this gene can increase the risk of developing PD and may lead to increased alpha synuclein production or misfolding. Second, LRRK2 gene. The LRRK2 gene is involved in regulating inflammation and autophagy, which is a cellular cleaning process. Mutation in this gene have been linked to an increased risk of PD and may lead to the impaired mitochondrial function. Parkin gene. The Parkin gene plays a role in the removal of damaged mitochondria. Mutations in this gene can lead to accumulation of damaged mitochondria, contributing to the neuronal dysfunction. Pink1 gene. The Pink1 gene is involved in the quality control of mitochondria. Mutations in this gene can disrupt the mitochondrial function and lead to the increased oxidative stress, which is contributing to the PD. So let's move to the next. The environmental factors, a complex web or influences. The pesticide and heavy metals. The exposure to the certain pesticides and heavy metals has been linked to an increased risk of PD. These toxins can damage neurons directly or indirectly by inducing oxidative stress and mitochondrial dysfunction. Lifestyle factors. Lifestyle choices such as smoking, excessive alcohol consumption, and lack of physical activity have also been implicated in PD risk. These factors can contribute to oxidative stress and inflammation, both of which can damage neurons. The ongoing research. Research is ongoing to understand the complex interplay between environmental factors and PD risk. Further investigation is needed to identify specific environmental exposures that may contribute to the development of the disease. Sorry. So here in this image we can see the manganese, copper, lead, mercury, and iron, and the neuron, the oxidative stress, the increased alpha sign aggregation, the mitochondrial dysfunction, increased ROS, and the motor and sensory impairment. The research at the future direction, a promise for hope. Parkinson's disease research is making progress in understanding the disease and finding new treatments. Scientists are focused on learning how the disease works, identifying targets for new medicines, and developing innovative therapies. So, I would like to call Dvij for to explain the conclusion. Thank you.
Thank you, Terry, for explaining very good. And yeah, so let's proceed with the conclusion and conclude this presentation. So, Parkinson's disease is a complex neurological disorder that affects millions worldwide. It stems from the progressive loss of dopamine producing neurons in specific brain region, the substantia nigra. This loss of dopamine, a neurotransmitter responsible for smooth muscle movement and coordination, leads to the characteristic motor symptoms of PD, such as tremors, rigidity, slowness of movement, and postural instability. In this presentation, we delved into the intricate mechanisms underlying PD, shedding light on the multifaceted nature of the deliberating disease. We explored how neuronal loss, protein misfolding, mitochondrial dysfunction, neuroinflammation, chemical imbalance, and genetic and environmental factors all play a significant role in its development and progression. Understanding these processes is crucial for developing effective treatments and therapies for PD. Current research efforts are focused on unraveling the complexities of these mechanisms, identifying therapeutic targets, and developing innovative targets that can slow down or even reverse the disease progression. The search for a cure for PD remains an ongoing journey, but with continued research and advancements in understanding the disease, hope remains for a future where PD can be effectively treated or even prevented. So I hope there's a cure for PD in the future. And by saying Salamat po, here we conclude our presentation. Just,
Thank you, ma'am.
yeah,
Salamat po.
thank you, ma'am. Salamat po. Good night
Thank you, everyone. Salamat.
for listening
Salamat.
PPT on Parkinson's disease. Thank you.
Thank you.